those of you starting, for sites that are watching, you need help with recruitment. But for sponsors watching, we do central campaigns. That's how we actually worked with Robert.
That's
Robert
right.
was our first sponsor, and he filled his study in how many days?
Oh, it
Like
was
a week?
quick. Yeah, it was. Yeah, it wasn't even a week. It was really, really fast.
Wow. So
Yeah, it was.
Robert Goldman, for those that don't know, he's affectionately known as the sponsor monster, but he's a global head of clinical operations for early stage biotech companies. He's worked with half a dozen at least, if not more.
Yeah.
Like foreign trained physician, not practicing in the US, of course, but he's doing what I think is better. There's less of him than there are physicians in the world. That's always a good barometer of like how well you'll do, I think, is supply and demand. Like if we just break down your career to supply and demand, how many of you are there in the world versus the alternative, right? Like
Right.
I also was a pre-med school major, then pre-pharmacy, and now I'm a site owner. And it's like, well, if you go by supply and demand, like site owners are a lot less in supply than at least ones with like decent sites
Yep.
than pharmacists or physicians. So for the youngsters watching, um, sometimes it's not, the traditional advice is not the best one necessarily for your career. But do you think you made the right choice? If
100%. 100. I wouldn't change it for the world.
Yeah, I never
I
actually asked you this.
Yeah, I mean, there truly. It's funny. I had lunch with a physician the other day, and he was telling me, he's like, hey, you could go and just do a quick residency, or you can go to some like rural states, and you can get a conditional medical license, and you can just then transfer it. Dan, I have no doubt I made the right decision. I leveraged the background. It helps me, in my day to day just by virtue of being a generalist, right? We've talked about that a long time. And so, whatever therapeutic area I'm in, it's, I'm gonna get it conceptually. So, not to mention, helping people on a global scale, bringing life-changing therapies and products. It's cliche, I get it, but it's really not. It's really my call to fame. So
Yeah,
for me personally, I made the right decision. Yeah, definitely.
I think you did too, but I'm not you, so I can't speak for you. But it's to me, it's just supply and demand. And glad you're here,
Thank
global
you.
head of ClinOps. See him at Save Our Sites. See him at. You're going to a bunch of other conferences. You're always
Yep.
going to a conference, man.
Yeah, I'll be on the circuit starting in February with Scope. Then I'll be at SOS. I'll be at some regional meetups.
You go to any
Um,
therapeutic area conferences, like
eh,
non-clinical trials, but just
I might.
like
I might.
DDW or OSCO or something like that.
Yeah. Not recently, but that doesn't mean I'm not going to start. I'm definitely
Mm.
going to be making a circuit, starting to work with some sites. Also through the consulting that I'm doing and things like that. So, um, very good chance you'll see me around.
Darshan Kulkarni, welcome, man. You're gonna be on these pods soon too. One day, you will make the show, Darshan. Uh, so, Robert, going from so us clinical research people, like especially at the site level or at the CRO level, we're very downstream from all the work, sometimes years of work it entails to get the IP, first of all, to discover it, then to get like commercially licensed by some entity that's going to take on the risk of R&D, presumably with the goal of it getting approved and then figuring out the sales part after, the commercial part after. So you're in this early stage where when do you typically get involved with a biotech? When they already have, I imagine when they already have an IP of some sort, or do they
Not
already
always.
you and say, "Hey, we need to find something"?
Yeah, so I mean, I have several experiences where, like one company I worked with had a preclinical stage asset, you know, but it was a very unique area in male fertility,
So what does
infertility,
that
I
mean
should
when
say.
they say preclinical stage asset? Like,
Yeah, sure. So basically, it's the time in which we're gathering the data that you need via FDA mandate to submit an IND package, right? So there's a whole bunch of different modules that the FDA requires in order for you to actually successfully submit an IND. And by the way, for the audience, please, it just irks me when people say, "Oh, my IND got approved." No, it didn't. No, FDA does not approve INDs. They either allow them to proceed or they place a clinical hold on the program. Those are the two outcomes of an IND. The only approvals that that happen really are NDAs, right? That's a new drug application versus an investigational new drug. And so in the preclinical phase, you're doing a lot of different, legacy things like, preclinical animal models, rat, bunnies, guinea pigs, sometimes, whatever is required from those discussions from the early interactions with the agency kind of outlines a clinical development strategy in terms of what sponsors have to do and what data package has to be presented as part of your IND submission. That's cytotoxicity, dermal toxicity, fertility, early embryonic development known as feed studies, you know, ocular toxicity, phototoxicity. Like there's so many different.
now is this for every IP that you want
It
to
is.
get to IND?
Yeah, yeah. It's very 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 common. To your point earlier, years and years of preclinical stage research goes into getting to a place where we can say, you know what, it's safe enough for us to dose our first human in a first in human setting or a phase 1 setting.
Yeah.
So you have to have extensive data packages, not to get too detailed here for the audience,
And
but
these are the mice studies and
it
generally mice studies, right?
they're Dan, some people don't want to hear the truth, but it doesn't do us any good not to say it. It can range from NHPs or non-human primates to canine models to to those rodent studies, and it really just depends on what therapeutic area you're working in and really what the requirements are of the agency during those early interactions dictate what type of studies you have to do as a sponsor preclinically and in what species that has to be done in. Obviously now we're FDA is moving away from the animal models and we're using a lot of what we call MIDD or model informed drug development, and you can do a lot of simulations and
Hmm.
use human data to kind of predict what will happen. It's really interesting, and I think AI is really helping to drive that transformation where we're not so focused on, well, let's see how it's metabolized in a rabbit, or let's see what happens if we expose, generations of mice to this particular product. How does it impact
This is
just
where AI does have some promise.
Yeah, I think so. I mean, because legacy way of doing this, it's you gotta, that's why these preclinical CROs exist, Dan. I mean,
Yeah,
I
yeah.
don't want to name any particularly. We can talk about it offline, but
Well,
the,
Darshan always talks about whooshy.
yeah, I'm not familiar with that one particularly. I mean, I've heard of them, of course, but I haven't worked with them. But I've worked
Mm.
with a number of those preclinical CROs who, their job is to help companies make, answer these questions, right? So
Yeah.
all of that data collectively goes into your IND application, and that ultimately is what gets it into the clinic. But to your point, it takes years and years and years of development. That's not even talking about finding an asset, right? Whether it's a small
Right.
molecule of peptide, gene therapy, cell therapy, where do you land? Are you in CBER, CEDAR? Where are you, CDRH? Like where do you belong, right? So you kind of there's such a
Do you
nuance.
think, well, you're gonna look for what's first? Like, are they actually looking for just promising drug candidates, or are they looking for something for a specific therapeutic area? Generally, which one comes first?
I mean, it could be both. It could be both. I mean, it depends, right? Because like in the context of medical devices, like your class 1, class 2, class 3 medical devices, or if you're looking at a general wellness product, or if you're looking at an actual small molecule to treat a disorder. Is it rare disease? Is there predicate items on the market that are currently approved for a different indication? You're seeking a new indication. I mean, it's so nuanced that it's a hard question to answer. I mean, typically companies are, there's companies out there, Dan, that are solely dedicated to repurposing currently approved drugs, right, for other reasons.
Yeah,
So
Christian
it,
Christian Angermeyer. I don't know if you're familiar with him.
Mhm.
Um, he's the well, he's the chairman and founder of Atai uh, Life Sciences, which is the DMT treatment-resistant depression. But he
uh,
has this fund, and he has the Substack I subscribe to, and he wrote an article recently. He said the top-selling drugs sold 36 billion in 2010 and were used by 112 million people. In 2020, the top 10 selling drugs sold 93 billion, but were only used by 18 million people. So
wow,
pharmaceuticals are becoming more precise and I guess less mainstream, although the GLPs kind of goes against that trend. But
yeah,
the trend in his argument is everything's becoming more like less of a mass blockbuster and more of a high cost specialty, sophisticated engineering and strategies and things like that.
yeah, yeah. I mean, it depends. I work for a company called Contraline. Um, we are a male contraception company. Who thinks of that, right? So we're actually
A
bringing,
lot of women.
well, but they wish that there was something that exists
Yeah.
that so it doesn't always fall on their shoulders. But um, you know, that's the whole thing. So, to drive the point home, when you're bringing in a first in class asset that never existed before in a therapeutic area, think GLP-1, right? The first GLP-1 to hit the market, it really wasn't that exciting because nobody really knew what the potential for that would be.
Right.
As it reached the real world, and obviously now there's cardiovascular protective natures around it. Obviously there's the weight loss part around it. It helps with your but there's also cons to everything, right? I mean, in terms of GLP-1s. So when we think of something like male contraception, for example, historically men have not very many options, right? They have the vasectomy and they have condoms, and they're not very effective. And obviously, the vasectomy is a permanent thing. Although people will say, yeah, well, we can reverse it. That has a very low success rate when you do reversals. So this will be the first time that an actual asset is going to reach phase 3 of clinical development, where men will have an option to control their own fertility in terms of not. And then again, there's a lot of women who don't do well with their female methods right now due to large side effect profiles. So you know it's exciting in the sense that it's a first in class asset and there's no predicate or something that even is close to it. um, and so kind of thinking about drug development in the sense that depending on what therapeutic area you're in,
Yeah.
how saturated is it? And then again, when you're working in a place like male contraception, people don't think of male contraception,
So your
right?
guys' example, and feel free to say I can't discuss this, but
Sure.
it falls under cedar, I would guess.
Yeah. Yep. 100%.
So all the headlines about the turnover there, does it affect you guys at all? Like you go from Tidmarsh to who's the new guy? The new guy is supposedly somebody that everyone likes.
Dan, it does. You would not believe the trouble that things. I don't want to say trouble. It has tremendously impacted
The turmoil
every
over
yeah,
there.
yeah, everybody's company. It's like, look, here's the overarching concept, right? Previously, you had a lot of senior leadership in the agency that they've seen it all. They're not very like phased by certain things that happen, if you will. And a lot of the senior leadership is now gone. So we're left with a lot of what we call junior reviewers.
Hmm.
And this is not I'm not looking to, I'm not, it's not a slight towards the agency at all in any way, shape, or form. It's just the reality. And so these junior reviewers, are obviously taking a more conservative approach because they haven't been exposed to what the more senior leadership people have seen, right? So when you're talking about, reasons why the agency thinks one way or the other, historically legacy way, you could go to a more senior reviewer and they could be like, oh, don't worry about that, it's all good, let it move forward. Whereas now we lack that, when I say we, the community lacks those more senior reviewers. So you're left with the more junior reviewers who take that, again, like I mentioned, conservative approach. And what that does is it impacts, clinical development programs. Oftentimes companies find themselves on, and again, I'm talking very broadly here. This is not specific to any company, but there's more clinical holds than ever in the past year,
Yeah.
due to various reasons, and so they're just taking a more conservative approach because
Do
that
you think
senior leadership.
ultimately that's good or bad for the industry? Like,
I think it's bad. I think it's really bad.
but
Um,
short term, but do you think long term it's good or bad? Like, do you think these junior people like kind of know what they're doing, or do you feel like it was better the way we had it before, or do you think that the FDA needed revamping a little bit?
I mean, I think it was better the way it was. I'm speaking strictly towards the FDA here.
Yeah,
Having the senior leadership who have been there, done that, moves things along. I still also believe that there needs to be a shakeup and there needs to be some alternative ways of thinking. But regrettably, that's not, at least in my experience, I'm not seeing the benefit of that right now because really what we're seeing is, you know, um, resources constraints. We're seeing delays in timelines. We're
yeah.
not able to converse. I mean, go get a type A, B, C, or D meeting. Good luck. I mean, like literally, you need to like almost lobby the FDA to get
Was
granted
that
a.
because of the government shutdown, or
I just think it's I don't think it has anything to do with the shutdown. I just think
huh?
it's the sheer number of bodies that are left in the agency that are able to,
Wow.
there's prioritizing like what we can and cannot do. So for example, if you've had a stall in your clinical program and you're requesting a type A and they know you've had one in the past, like you're not going to get another type A.
And can you break those down? The type A, B, C, D. What?
Yeah, so I mean a type A is, just kind of a when your program is stalled. You can look it up. It's very very simple. Type C is more along the lines of like then you have your like pre-negotiation meetings, which is probably closer to a B, and then C is essentially like I need guidance on clinical development, future like forward looking. For example, if I want to get alignment on a phase 3 design or a phase 2 design, you can ask for a C. But yeah, I mean, just
Okay.
look it up. They have a lot of tables and a lot of resources out there.
Okay.
But I'm very, very, I use A lot and C quite frequently in my personal experiences, which might be different from other people's. But I'm just saying that to say, I think that generally speaking, right now, they're being very careful with handing those types of meetings out. And if you get one, you better really make it useful, right? Have your questions really organized, be very pointed, get the most out of it because the idea that you're going to get another one is at this time is probably very unlikely.
Unless
So
you have one of the breakthrough designation, fast track
no, even
designation.
even that, Dan,
Even
even
then,
that,
okay.
even breakthrough designation. I mean, breakthrough is something that's typically, at done prior to phase 3.
Yeah.
Fast track is, also those different designations. They don't guarantee you like a meeting cadence that's any different than the other type of applicants out there. So having those designations just really make it easier for your submission standards and able to roll in and submit things on an ongoing basis as to opposed to waiting to the end and things like that.
Okay.
So it streamlines things a lot better when you have those designations, but it doesn't afford
Okay.
a more frequent meeting cadence than somebody else who doesn't have that designation.
Okay. And that's a good segue because so when you're planning a trial,
Mhm.
let's say you can skip phase one for whatever reason, you have enough data from the past. Maybe it's a repurposed drug or something. So you go to phase two, right?
Mhm.
Um, you're testing it on the target population that is essentially your hypothesis. So where do you start? Do you look at previous trials done? Do you talk to the FDA first, or do you have to plan it and then talk to the FDA, or how does that, like, where do you start when it's time to
Yeah.
start?
So typically you're gonna, you obviously have to plan. You have to have a protocol, at least a synopsis is ideal, to pivot into a phase two. And, really the statistical methods in which you're going to show what your endpoints are mission critical at this stage. So your primary endpoint, your primary objective, your secondary endpoints, your secondary objectives, and then of course
Okay.
your exploratory.
And
What
how
am
do
I
you choose
trying?
those? How do you choose those? Like, how do you guys decide on those?
Very carefully with an alignment with FDA and a very strong statistical team, right? A lot of
Okay, so FDA
biostatisticians.
FDA is involved when you're
Oh yeah.
deciding endpoints.
Oh, absolutely. Absolutely. Depending on your
Okay.
therapeutic area, you have to have alignment on that so that you can move the clinical program forward, right? From phase two to phase three, and then
Okay.
even the two A's and two B's in between the phase three if it's there. But at that point, you may not even know what your LD50 is, or your lethal dose at half the amount of the most highest dose of what you could administer somebody. So you're kind of, it could be dose ranging, like we don't know the best dose, and we're kind of, because FDA wants you to use the lowest effective dose to achieve the same effect. So you do that with pharmacokinetic studies, pharmacodynamic studies, which is known as PK and PD studies. And we're trying to determine not only safety, efficacy signals, establishing a safety profile, but looking for efficacy signals. But we're also trying to find what's that dose in the context of a drug that we're going to carry forward into phase three. So these are the questions you're trying to answer in a phase two setting, at the same time having very focused endpoints that will enable us to have an end of phase two meeting with FDA to say, hey, here's the data. And that's where a lot of drugs fail, right? A lot of compounds and therapies fail at that place because it just isn't as good or better than what's already currently on the market. And so you have to demonstrate at least non-inferiority to what's already currently approved in any disease indication. If there's already something out there that's approved, why would FDA approve something that doesn't work at least as good, if not better, than what's already available? So there's a risk benefit ratio that you really have to do an analysis here as a sponsor to decide, how to move things in and through the pipeline to ensure that there's value there. Because as you know, it's millions and millions and millions of dollars to get things across the finish line, and even
So
from phase to phase.
so is it like in those 505B, what are they called, the when you're repurposing
505(b)(2). Yeah,
B2, right?
505(b)(2). So that's an ANDA. That's an abbreviated. So the definition is so essentially like an NME, right? So a new molecular entity or a new chemical entity. The definition of that is a compound that has never been approved by the FDA either on its own or in combination with another product. So
Okay.
that means it's a NME in the true sense of an NME. And I wish we had Darshan here because this is right up his alley. I'm sure he'll correct me if I'm going wrong here, but I'm not in this instance. Um, and then so the moment any a compound is either approved on its own or even in combination with another product, even in a different population or a different therapeutic area, it now becomes a 505(b)(2), which is now considered technically a generic, and you're now going down an abbreviated pathway, which means hey, FDA found that compound, whether again it was by itself or in combination with another, safe enough and effective enough to be approved. So sponsors can lean on that simple fact that hey, this drug we know it kind of de-risks it a little bit, and that's why you're
Yeah.
seeing a lot of pro drugs are popping up on the market
Right.
now.
Right.
Because it's like, well, FDA already approved it, so we know it's safe, we know it's effective. Now, what we're intending to do with it is a completely different
Right.
discussion, right?
Different use case. So, but
Yes.
the presumably you don't have to do all the extensive safety study as long as you're bracketing it within the area under the curve or something like this,
Well,
right?
not always, because people what you're referring to, Dan, is something called right of reference.
Okay.
And so, you got to be careful with that. And again, I mean, Darshan would be a much better person to talk about this than myself. But when you're trying to like look at the other IND or NDA holders' safety data and leverage it for your own purposes, um, again, like you don't necessarily need to do that, because what you know is that FDA already approved this compound. So they've already found it safe and effective. So what
Okay.
it does is it kind of defines the safety database that you're going to have as a sponsor to
Okay.
bring it to a new therapeutic area. So,
So you just have to
again,
prove like it's just as safe as
right.
these other drugs that were approved, even though we're doing a different formulation or method of administration or
Right. Or
whatever,
just
right?
a completely different therapeutic area. I mean,
Uh-huh.
something that comes to mind is like Skyrizi, for example. You see all those Skyrizi commercials, right?
Yeah.
Or like Humira, or all those different things. So these drugs were all, they came out for psoriasis, right? Psoriatic arthritis, psoriasis,
Hmm.
and then they realized, well, hey, this same drug can be used to treat irritable bowel syndrome, Crohn's disease, things like that. So it's interesting because that's a different. It's the same API, it's the same active pharmaceutical ingredient, but it's interesting, at lower doses it works here, and then at higher doses it can start having impacts on the GI system, right? So
Hmm.
your integumentary system, your skin, your dermis, is the largest organ in your body, obviously, right? And it's amazing that at a lower dose, in the context of this one drug, you're focusing certain receptors that impact your immune response that creates psoriasis. But then on the other hand, when you increase it and you infuse it, not subcutaneously, but infuse it directly into the bloodstream, now you're getting, GI benefit. And now they can run a study to demonstrate improved safety and efficacy for the treatment of, Crohn's disease, which is another an autoimmune disorder. So, this is how things move along down the pipeline, but like one drug is now treats multiple conditions. And so typically on the early phases, large companies who have a lot of money, they can really, kind of explore 5, 6, 7, 8, 9, 10 therapeutic areas in a preclinical setting, even do it in the phase 1 setting. And then based on the results of those studies, they narrow the funnel to say, okay, well, we're seeing positive signals in this area. We're going to move it towards that direction.
Right,
And
right.
then they chop everything else off. So
It's just
that's,
like Manjaro or tirzepatide. They're
yeah,
testing it now for, or they're talking about testing for depression, Alzheimer's prevention. Dave Ricks
heart
was
fa-
on, and he was talking about
exactly.
this.
They're talking about it for stroke prevention now, and
Yeah.
heart failure. Like if you had heart failure or peripheral artery disease, or with or without diabetes, and or you had a heart attack in the past or a
Hmm.
stroke,
Hmm. Hmm.
like there's yeah. So I mean, this is exactly how, Dan, because what's happening is now the drug's in the wild, people
Yeah.
are using it for all types of things, and we're seeing,
So
um,
presumably, okay, so this is where the academia is valuable because they publish, right? They're always publishing. Like scientists in the lab are usually finding things on accident and then making a publication somewhere, and then it takes like a business person to look at it and say, well, wait a minute, maybe we can acquire this, or like Darshan said, do an in-licensing deal. I'm assuming with
uh
that university or with the tech transfer office there, and get this thing into get a protocol, get an IND, get a protocol in that order, right?
yep, that's right. Get a
Okay,
protocol, well, I mean, you can't have an IND without a lot of preclinical data, right?
but you can use what's out there if there
Sure,
is, right?
sure. I mean, yeah, there's, again, I'm not a regulatory
Yeah,
strategist, but
yeah.
there are ways to, leverage what's out there to, make a case to pursue another therapeutic area or indication.
Got it. And you've been involved in these kind of processes before, or you usually get involved a little bit after this?
So, me personally, I've been involved on both of these types. So I've
Okay.
been involved in the preclinical aspect, moving, assets from preclinical into the clinic. And I'm also involved in, moving things from phase 2 to phase 3. So
Man, good thing you didn't become a licensed US. You'd
yeah,
have been one
yeah,
of many as opposed to like one of a few,
yeah, it's
literally.
true. I'm very fortunate to have the experiences that I've had, and I'm grateful to all the companies that, gave me that shot. So
Now we're
for
gonna
sure.
get into how to design it and stuff like that. We'll have you on maybe a series of podcasts, but how does one even get to do this? I'm guessing you kind of, because you got in at, from ClinOps, I'm guessing you like reverse engineered your way downstream by accident or upstream by accident. People kept asking you probably, hey, can you help with this? Can you help with this? And pretty soon you're like getting close to like the bench research at some point, right?
Yeah, I mean, you're talking about like how did I like my career progression, or
Yeah,
what are
because you
you?
were a CRA at
Yeah,
one point. Not that
yeah.
many
No, I mean,
CRAs get to
Dan,
even know about this stuff.
I was a project associate at INC Research.
Yeah, so
I
how do
mean,
you just like real quick, like the Cliff Notes version of what
yeah.
took you down this path? Was it intentional or was it accidental?
I mean, listen, it's I think both.
Yeah,
I think it's definitely both. But
as
look,
with
I
all
mean,
things, it's
yeah,
yeah.
it's grind, it's determination, it's grit, it's doing more than you're asked to do, it's
Mm.
standing out. I mean, that was just
Adaptability.
yeah, that was just always me. I always wanted to do more than I was asked for. When other people, didn't do things, I would be
Now,
the
did
first
they
one to
ask you?
volunteer.
I'm assuming a lot of them asked you because you're a medical doctor, and so
Well, I think they understood that, the medical background certainly makes you a generalist in the sense that, like,
yeah,
I'll go, I mean, I can go, I'll give me an SOA and give me a inclusion exclusion criteria, and I'll go monitor any study. It doesn't matter to me.
yeah. But
You know what I
even
mean? Like,
doctors, man, even so, I work. I work with a handful of different specialists here in Yuma,
yep,
and I also invest in different biotech. So sometimes we have discussions. We don't invest. I have a rule. I don't invest in what we're doing studies on. But I talk to my doctors because, like, they're doctors, so I figure they gotta know more than I do. Let me ask them. So I asked my pulmonologist about these cancer results, and he was like, "What is this?" Like, he didn't even know
right.
what ORR was.
Yeah,
He asked me, and I said, "Well, I'm asking you because you're a doctor. Like, don't you know?" He's like, "No, we doctors don't know anything after medical school. We just
it's true.
specialize in what we
It's
do, and
true.
that's it."
It's true. It's true.
So for you to be more of a generalist this way, you had to constantly feed your mind with new stuff on your own with no one telling you. Like, you're probably reading PubMed
All
here
the
and there.
time.
Like, no one's
Yep.
telling you to do it.
Need them. Lived in the New England Journal of Medicine to understand what's the newest and greatest. And
So do you
yeah, I
still
mean,
do
Dan,
it, or are you too
I
busy
oh
for that?
yeah, no. I still do it. I'm a sponge, and I yearn for information, and
So that
I'm
stuff pays off later on down the road.
I think of myself as a lifelong student. That's
Yeah.
really what I like to think of myself as, because the moment you start to say to yourself, all right, well, I know enough, or I know everything, I
Mm.
promise you, you don't. Like,
Well,
I don't know everything.
most people don't. I would say most people don't say that. Most people say, "I'm too busy and I got
Yeah,
to focus on what's paying my bills." And then technology just advances quickly. If you take 5 years off of some therapeutic area, you are behind on,
yeah,
especially something like oncology. I mean, you are behind. You're in the Stone Age at that point.
yeah. That's true. That's
Okay.
true. Yeah. So I mean, and look, it's I grind. I would just, grind. I was living, you know, not the most greatest life, as a CRA road warrior, and
Mm.
rose the ranks of all of those different positions all the way through principal CRA. And then, I was offered a study startup lead. And I'm like, well, wait a minute. Why do I want to be a startup lead? I want to be a CTM. That's the natural progression. But I checked my ego once again at the door, and I'm very grateful that I was a study startup lead because now I know startup as well, better than most people know startup, right?
Right. And these
And then
scientists coming up with these things, or these regulatory specialists, they have no clue
no,
like what the real
they're
problems are at the site level and why studies are
right.
delayed and all that stuff.
Exactly. So I think
Mm.
because of all of those different roles that I held at different organizations, I've been able to kind of combine everything and create my own playbook, if you will. And so
Mm.
it for me, it's proved very beneficial. And I think that's where, colleagues that I've come in contact with decade or more ago, reach out to me till this day and, you know, offer me different opportunities. Hey, this, that, and the other, because they remember that work ethic. They remember that yearning for knowledge and curiosity. And, I think that goes a long way. And then of course, but nothing replaces anything but hard work. Like it's hard work. You have to be committed, and it's just you can't there's no entitlement here, right? Just because you've been in the industry 25 years doesn't mean you're entitled to a certain position. And just because you have certain letters after your name also doesn't entitle you to a certain position. um I've worked with brilliant people that don't even have a college degree. And I've worked with PhD level people that the non-college degree holder will run circles around.
Mm.
So, I'm just saying, the industry itself now, because, during COVID, they overhired and things like that. I think they're
Yeah.
becoming a little bit more, stringent, but I don't agree with that approach. I think there's a lot of really, really, really well qualified people out there that, I mean, Dan, for me, real world experience trumps anything you learn in a classroom.
Well, I was
So
going to ask you, what's harder to learn? Like study ops. So once a study's going, like how to have a successful clinical trial, at least you can't control the results, but at least have a
of course,
smooth study, right? And
yeah,
on budget, on time. What's harder, figuring that out or figuring out the regulatory pathways, maybe the proper endpoints and all that, like more scientific stuff? Which one do you think is harder to master?
I think that the operational execution and delivery of a study is much harder to learn and master because it simply cannot be taught.
Huh.
Um,
And
you
the other
know,
stuff can be taught. If someone's curious enough, they can
yeah, I
figure
mean,
it out.
look, stats is stats is difficult, absolutely. I'm not minimizing it at all, but status it's once you've figured it out, it's pretty
Which
pretty
one? Which
straightforward.
one can AI take over faster? Stats or the ClinOps?
Oh, stats for sure. I mean,
Yeah,
that's a
that's a
no-brainer,
leading. That
right?
was a leading
Yeah,
question. So
that's a no-brainer. Yeah,
yeah.
definitely. So, it takes a lot of finesse to ensure, like what you described, that things get done. And I'm the first one, like AI will never replace operationalists. It's just not possible.
Mm.
It's not possible.
It might replace certain aspects of the operations,
Sure, sure.
but not
But
holistically. The kind of like this conductor of the
yeah,
orchestra, right?
you're not going to have the Optimus, robot. And I'm a huge
Elon. Yeah.
Tesla and Elon fan. But look, like no matter what, you're never gonna have one of those robots do my job. It's just not possible. Some people argue and say it is possible, but I don't think it's possible. So you've really got to, when you're delivering and executing a clinical trial and thinking about planning it and all the things that could go wrong and all the risk mitigation things, and when you do this and when you do that, it's very, very, very nuanced. And you know the technology today will tell you that, hey, there might be a problem. You need the human to determine, is it really a problem? So like signal detection, risk detection,
Mm-hmm.
they're surfacing all these things to you that, based on the algorithm, it's doing its job. But there's
Mm.
cases out there where half the time it's like actually not a problem until it is a problem. Now,
Yeah.
that doesn't mean I don't want to know about it. And legacy way of doing things is more manual, so now I know about it sooner.
Right.
But still, like, you need that person in the middle to
Yes.
really move things.
And
Yeah.
thing between about ClinOps that depends, like the levers to pull are a lot more psychological. So if you have a site that's not entering data fast enough or not accruing fast enough, or maybe you have a CRA that's not checking SAEs or deviations quickly, right? Or not catching them. That's fundamentally like a still a human issue. That there's a lot of psychology there that you can
Yeah.
pull, like those levers, versus a biostat. It seems like it's very binary, or like a regulatory affairs role. It's like very binary. Like this is the path. There's is no other levers to pull really. But the solutions from the cleanup side generally come from the people. And
Yeah, and the solutions come from experience, right? The solutions come from been there, done that, and being able to think outside the box and really, guidance is guidance, right? Like FDA wants to see this. And so again, we shouldn't, again, we should not minimize how important, the statisticians are,
yeah, yeah.
and how, clinical development strategists and regulatory affairs, like there are a lot of products out there where you need to have a very savvy regulatory strategist who
Mm,
knows how to navigate and weave in and out of the framework that's already in existence to get any forward momentum and progress. It's not as straightforward as we might be making it out to be,
mm-hmm,
but it's still, that's why there's like such synergies between the two. I would call them crafts. Like it's an art, really. In order to get something to move forward is an art. In order to actually execute it in the clinic is an art.
and that's where
And
you kind of spend most of your time, is in between these two crafts.
yeah, yeah, I spend a lot of time with, brilliant regulatory strategists, and I spend a lot of time with, great operationalists. And there, and then of course, you have to get it across the line, right? To your
Yeah.
point, within budget, within scope, data quality, integrity, holding people, accountable, and making sure things are moving and catching things before they become problems. So that proactive mindset versus that reactive mindset is really critical to the success of what things are, of how things are moving along. Yeah.
Okay, so when you get to protocol time, right? Designing,
Yep.
let's say you found the endpoints, you found the primary endpoints, you guys agreed primary, secondary, and then exploratory endpoint, which I don't know, I don't know if you're a fan of exploratory endpoints, or like what's your
Well,
philosophy on those?
Well, you got to be careful with those because you might find something out that you you probably would have not liked to have known about. So historically, you got to just be careful to really understand, is the you know when you ask a question, just understand that you might not like the answer you get.
Mm-hmm.
And so that's really how I think about exploratory endpoints. Um, actually with everything, really. I mean, it's even with sub studies and different cohorts and different, stratification factors. If you start to implement things, just always remember the other side of the coin. You might not like what you see, and it could create more questions than answers. So it's really a fine balance of understanding what the impact is of asking a question or including an extra exploratory endpoint. Um, for example, I don't know, a lotion dispenser or a gel, just some sort of dispenser, right? Well, there's people out there that'll actuate it one time and maybe the dose coming out is not the exact dose we thought it was. And so, you know, you might start to do an exploratory endpoint on accuracy of the dose dispenser as an example. I mean, that's probably a really bad example,
Mm-hmm.
but what I'm trying to say is you have to be
I think
careful with what
Darshan
you
brought
ask
up the
for.
same example on a fraud pod recently.
Yeah,
You
yeah.
gotta check
So
what's going on with these dispensers.
you got to check, and so what I'm saying is if you have an exploratory endpoint around the success rate of these dispensers and they're not going to fail, and then all of a sudden you have a bunch of failures, now, like, no matter how good the compound is inside that dispenser, if we're not dispensing an accurate dose every time, each time, consistently, then is the data valid anymore? Probably not. So
Yeah.
small things like that. So I guess the overarching thing I think for me when it comes to exploratory endpoints is just, you need to use some caution because you might not like the answer you get, and you need to think that through. And that's all done with risk benefit analysis, right?
Okay, so then how do we decide on, let's say, a phase two? Phase two generally safety and then some efficacy.
Hmm.
Phase three efficacy and then safety as well. Let's say phase two, how do you decide on how many visits is the study,
Yeah,
your n number of patients? Like, how do you decide those things?
yeah. Well, like I said, Dan, it's really the engagement with the agency who kind of help you determine. Agency's not going to tell you, what dose to use or how many visits to have and things like that. But in order to meet the goals and outcomes of the study, whatever those may be, you have to settle on how many visits and you have to settle on what the N is. And again, most studies are what we call statistically powered to meet to have statistical significance. And, not getting too far into stats, but there's something also called the p-value. And the smaller the p-value, the more statistical significant your data is. So you have to have enough power, enough N, enough participants in your study to show that we have a statistical significance, because an N of one doesn't really mean much. So it's a very complex situation when you start working with, what do we need to show at this point? How much of the data safety database do we have to, assume to? And then how much more do we have to, finish off in the phase three? And so it's a really fine choreographed dance between how many visits there are and how many patients there are, because you want to have enough oversight, you want to protect the safety and welfare of the patients, and you have to satisfy the regulatory bodies, and you have to satisfy the IRBs and ethics committees. And so making sure you're doing all the right labs and you're doing all the right check-ins and you're doing everything that you can do to preserve the safety of the participants who are so gracious to give their time to participate in the study, really drives the cadence of how many visits there are and what has to happen at each visit. It's not because sponsors set out to make things difficult. They really set out to, I mean, some do, some try to
Yeah.
jam a whole bunch of things into a single study to answer a whole bunch of questions. But, ideally, you really need to think thoughtfully around, what you want to answer, how you want to answer it, and when you want to answer it in order to inform study design. To your point, how many visits are there? How many patients need to be in this arm of the study and this phase of the study? So obviously, as you know, I mean, phase one is very small, phase two is a little bit bigger, and then phase three is your large intent to use population, which you have to enroll enough patients to meet the exposure for the safety database that the FDA will tell you, what you need to achieve. And whether that's, X amount of people for 6 months, X amount of people who had at least one dose, the remaining amount of people who had up to 12 months of exposure. And that's how you kind of inform the study design at a very high level. Again, it's much more complicated than that, but
Yeah. No, who
that's
designed, like, you come up with this generally? Like the ideas
so
for the numbers, or
it's in collaboration with, lots of different stakeholders, your regulatory team, your biostatisticians, your scientific officers, and your scientific people. But ultimately, like from a phase two to a phase three, you're not fundamentally changing too much of your design when you're at a phase two stage. Phase three is just recapitulating what you did in phase two with maybe some optimizations and some efficiencies that you've learned from phase two that didn't really work out, assuming your asset moves to a phase three. So once you settle on a design, Dan, you really don't want to stray too far away from the current design that you have. Of course, I'm not saying or insinuating you shouldn't make efficiencies where you can, or you shouldn't optimize where you can to reduce patient burden and site burden, because what we saw in phase two, maybe we were expecting to see some lab value that we never saw in phase two. And so we were testing it every, 2 weeks. Well, now we don't have to do that because it wasn't an issue in phase two. So now we can kind of decrease the cadence when we test for that specific marker in phase three. But the idea here is that phase two design is not very different than phase three design. It's just a larger scale study, maybe your endpoints. And again, when you have that end of phase two meeting with FDA, that's really the time to align on, okay, what does the agency need to see so that your product gets approved? And you align on your endpoints, which might differ from phase two. The sample size is absolutely going to differ from phase two. But really the design at the heart, right, is going to be
Mm-hmm.
very similar to phase two, actually. And you're going to narrow down one dose, whereas in phase two, you use multiple doses. But again, it
Yeah.
depends on the therapeutic area. Nothing in research is static, right? Everything is always, it depends.
Everything depends.
Yeah.
Do you have like I guess a table or cheat sheets for at least a reference point for where to start? Like if I have a study that we're gonna do, well, you're always aiming for double blind randomized whenever you can, placebo controlled, but let's say if you were to do an oncology, maybe single arm is acceptable. Do you have like different categories for, okay, I'm running this kind of trial, this is my past trial experience, or these are like competitors that have done this? Are there like categories? Is it that simple, or that I guess uh, stratified, or are you kind of like freewheeling it the whole time?
It's really, I think it's freewheeling in operations. It's not, I mean, obviously you learn things as you progress and,
Mm-hmm.
what works, what didn't work, what worked okay, and what worked great. So for me, I don't think there is a cheat sheet or a checklist. Obviously, in, rare disease and sometimes in, even in contraception, right? You can't do double blind studies, right? I mean,
Right, right.
they've done them in legacy like female oral contraceptives they have, but certain instances you have to do open label studies. So, but the
Is that
idea
because
behind,
it's unethical, or
yeah, I mean, well, yeah, it could be even in
okay?
like rare disease, right? I mean,
Yeah, yeah,
if you're working with a disease population who there's only like 50 patients in the world who have this,
yeah,
why do
yeah.
why do we need a double blind? No, you don't, right? You
Well,
just
I
need open
understand. I
label.
understand for okay, open label, but placebos another story, right?
Yeah,
Um,
placebo's a totally different story, but I mean, when we talk about double-blind, randomized, controlled, placebo-controlled studies, that's
mhm.
the reason, right? Because you want to have your control,
Yeah,
and your
yeah.
control is your placebo to make sure that the drug is actually working as intended. So you have to have that. But a lot of the studies, will have that double-blind phase, but then after some juncture of time that goes by, most of those patients will have an opportunity to get active open-label drug,
Hmm.
and they'll never know whether or not they were on placebo or active because it's double blind, and you don't know that information until the blind is broken. But
Yeah.
in terms of development from an operations perspective, the playbook is not like it, you don't execute a trial because it's open label differently than you would if it was double blind. I mean, there's different challenges, right? Retention in an open label study is presumably going to be higher for sure because the patient's getting the drug. Now, whether or not it's working for that person is a determining factor on if they stay. Whereas in a double blind study, the patient knowing, well, I might have placebo, I have this percent chance of getting placebo, they might not make it through that entire phase where now I know I'm actually getting active drugs. So the playbook is not, it's not, for me, I think that's why this collateral doesn't exist today,
Hmm.
right? Like there isn't a checklist or there isn't, but it's just kind of it's a learned lesson in terms of, the type of study you're running, the therapeutic area in which you're running it, the population you're working with. Are you working with elderly, young, minors? Are you working with adolescents? Are you working with, who are you working with? Men, women? Like, who are you working with, right? All these
Hmm.
different variables that input to the way you run a study operationally matter. And so you can't create some like, playbook to do things. Well, if it's this was type of study, you're going to run it this way. And here's the checklist for it. You kind of have to think outside the box because regulations are changing daily, right? Guidances are being updated all the time. And to your earlier point, that's why you have to keep reading and you have to keep educating yourself and attending webinars and going the extra mile and reading books and attending conferences so you can learn and stay up to date with the newest and greatest. Because
Right.
look, I mean, if we look at the difference between ICH, E6R2 versus R3, there's some significant changes that impact the operational execution of a study and what sponsors are beholden to, what IRBs are beholden to, what investigators are beholden to, what vendors are beholden to, what any service provider is beholden to. Like they don't even, so like this is fundamentally very important to understand.
Hmm.
So that's why it's difficult to create a checklist or a playbook because it's just an ever changing dynamic.
Now, what about these like regular meetings with the FDA? If you can get them, like, presumably when you plan a protocol, you're talking to them, right? At least once
Yeah,
prior.
yeah, there's early engagement meetings, pre-IND meetings, that you can get kind of their, are we on the right track?
Okay.
Are we a biologic? Are we a drug? Are we what are we, right? Do they concur that we're in this
Okay.
bucket, if you will?
And then when you
So yeah,
design this protocol, do
yeah.
they usually look at it and say, "Okay, we're in agreement with this," or do you not always get that luxury?
I mean, I mean, you don't always get that luxury.
Hmm.
Sometimes people do things um, at risk in the terms of like, you know, FDA will acknowledge, but they're not going to tell you like, oh yeah, this is great, we
Right.
agree.
Right.
Like they may agree, like they might say they agree, but they're not going to back themselves into a corner to where you can move forward with some sort of assurance that, oh, as long as there is something called a SPA, a special protocol assessment, which is typically done between phase 2 and phase 3. And if you have a SPA going into phase 3 and first of all, you have to meet the criteria to get a special protocol assessment, assuming
Okay.
you're granted
It's
this.
not automatic for
No,
everyone.
no, 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 no. Assuming you get that SPA designation, it affords you like, you do have a, as long as you've hit your endpoints and you've done everything you previously agreed to do with the agency, you have a very good chance at getting an approval, right? But
Okay.
still, nothing is guaranteed slam dunk, even with a SPA, because it depends, right? What happened throughout the conduct of that study? You start looking at your intent to treat populations and your per protocol populations and how disparate are they, and they start looking at, well, how well was the trial executed and how good was the sponsor's oversight, and then that trickles down to your CRO partners, your service providers, your site performance, your how many deviations do you have that impact safety and efficacy, how many patients belong in this
Hmm.
data set versus that data set. And you have to have enough of an N and enough of a power to show that statistical significance that it's compelling evidence to show that this, we did our part and we've met this, and here's why we've met this. And that's where the stats part comes in, which is, it's a very complex process, but that's why all the rules, how are mission critical, and working collectively and closely with each of those people throughout the entire clinical development pipeline and strategy is, like I mentioned earlier, a really well fine-tuned choreographed dance. And listen, you never know what you're going to come into. You might expect or anticipate this, that to happen, but what you anticipate and expect may never happen.
Hmm.
And something completely opposite may happen. So you have to be able to pivot very quickly and know which direction you're going to move, when those types of things surface.
Do you start designing the phase 3 protocol even before the phase 2 is over? Like, let's say you have interim
Some
results.
some people do. Some
Yeah.
people do. Some people do. I mean, again, it depends. It
Do
just
you
it
do you
just
get approval from FDA to go from phase 2 to phase 3? Is that, or are
you
you
you do. I
okay?
don't know if it's an approval, but you have what at the end of phase 2 you have what we call EOP2. So the end of phase 2 meeting, and that's really your opportunity. You submit your CSR, your clinical study report, your entire data package to the agency, and then you have an EOP2 meeting with the agency,
Hmm.
and they decide based on your clinical study report whether or not the asset can move forward to phase 3. It's not an
Okay.
approval per se, but they either will put your asset on clinical hold, or they'll say
Mhm.
they'll meet with you at EOP2 and they'll say, okay, everything looks great, and they'll help you align on the
I see.
endpoints, the sample size, which allows the asset to move forward to phase 3. But you cannot move an asset into phase 3 without FDA interaction in the context of an EOP2 meeting.
But that's mainly from a safety standpoint, right? They're not really looking at efficacy there.
They're looking at both. I mean,
Okay.
because they're looking at both, but primarily it's safe and there's enough efficacy signals to demonstrate that it's working
Okay.
as intended. And then in
Yeah.
phase 3, you're really demonstrating on a large sample size
Yeah.
that
Because
the drug is not only safe but efficacious.
I would imagine it gets more nuanced when, like, if you have a first in class or first time being developed, first of its kind therapy, it's a little different as opposed to like if you're number 10 down the list of another Me Too.
Absolutely.
The FDA is going to probably hint at or tell you like, hey, standard of care is, we're expecting. They may not outright tell you, but I'm assuming management would know or executive leadership would know from the sponsor uh, what they would consider successful before trying a phase 3. And a phase 2 is where something like 80% of trials go to die. Like they don't
Yep,
make it past phase 2, right?
that's very true. Yeah,
So who decides that? Sponsors or FDA, or like we're both?
I think I mean, it's both. It's both for
But the
sure.
majority of it is the sponsor themselves, right? Like, oh, we didn't really reach what we feel
Yeah,
comfortable
I
with.
mean, because it looks you can't argue with math, right? Math is
Yeah.
very objective. You know what I mean? So
Okay.
you either met your endpoints with a statistical significance, meaning a low p-value,
Yeah.
something like 0.05 or less,
Yeah.
or you didn't. I
Yeah.
mean, it's
Yeah.
it's really that simple. I mean, I
I
know it
see.
sounds simplistic, but if you didn't meet your endpoints or your study goals or objectives, I mean, why even proceed to an EOP2 meeting, right?
Right. Right.
Maybe you would in some instances to kind of, get guidance on, well, if we go back and do another phase 2 and we
Uh
do
huh.
things differently, like you have to do a retrospective analysis of why you didn't meet those endpoints, right?
And that's where
And
they'll
then
put out some press release like, hey, we're internally looking at our results, and
exactly,
investors start
exactly.
freaking out and all
Yeah,
that
and
kind of stuff.
you got to evaluate whether or not, like what was the root cause of why? Like, is it, I mean, Dan, I'll be honest, like drugs don't make it to phase 2 and die because it doesn't work.
Mm-hmm.
Like the compound itself, it's probably a lot to do with, the innovators, and I've said this at SOS, and I'll say it again at SOS again next year. It's like the innovators in this space are really by far and large the small biotechs, right? The small
Right.
biopharmas.
Right.
And then you have the big guys come in and acquire. I mean, J&J just acquired some company for billions of dollars, for some asset in I can't remember the indication, so I'm not gonna
Yeah. Yeah.
speculate. But
Okay.
my point is that,
Huh.
the innovators, the true innovators are really the smaller players that just don't have the
Of course.
capital,
Of
the
course.
resourcing
But you
to.
really think like with that 80% or whatever that fail phase 2, you really think it's not due to the bad science? It's due to poor operational execution?
I think it's multifactorial. It's not one or the other.
Right. Right. What
Because
what
again, it's like,
percentage would you attribute to science? Like, hey, this just there's nothing you can do here. This is not going to work.
Dan, I don't know. I
I'm putting you
mean,
on the spot, man.
I
But
know
is it 80? You're saying it's not 80.
I mean, I don't think the science. No, I don't think it's because of the science. I mean, I just think that the
Okay.
program, the way it was designed, the way it was executed, the way that the data was collected, did
Interesting.
not demonstrate the full capability of what that asset is actually capable
Well,
of producing.
that's the last thing investors should be hearing because they're always clinging on to like these companies that are not going
Late,
to do anything is because
yeah,
you just gave them more ammo to like hang on because they're the science, it's not the science, it might be the ClinOps. We'll just do it again. We'll dilute the stock again, run another trial. this time we're going to fix it. And then I don't know how many of those. Somebody should do a study. Maybe Ken Getz did the studies on this.
Yeah,
How many of those make it past phase two?
yeah. Great questions. Like I said, I mean, because my whole hypothesis is that if it was the science and the compound itself, it would have never made it out of, like, it would have never gone as far as it's gone. Like when you complete a successful phase 2 study or a phase 2B study, and again, if you met your endpoints, great. But also, Dan, like, regrettably, sometimes you're doing dose ranging studies in phase 2 and you just can't find that optimal dose. And no matter
Yeah,
if you titrate up or you titrate down, like, you're not getting a signal of efficacy, even though it's
yeah,
safe.
yeah,
So the product dies. So like that, in that case, it's not an operational issue. It's a science issue, right? It's a formulation issue. Maybe it's not as
yeah,
specific
yeah.
as we thought it was. So I mean,
But
it's
sometimes
very
on the other side of the coin, if you do it, you do a crossover study. Well, we didn't get enough time to show that this dose was the optimal one or enough of an N. So that's
right.
poor study design in that
Exactly.
case.
Exactly.
Yeah.
100% nailed it.
Yeah, it's
Yeah.
complicated stuff, man. No wonder
Yeah, it
investors
is.
keep losing money on these things.
Well, it is complicated, but that's also why, it's it, it's complicated, but human data is very important, and
Of course.
very, the investors these days are very savvy, especially in the biotech life sciences space, and
Hmm.
they know what to look for. And at the end of the day, it's, there's just, I think every stakeholder that's involved in development of an asset, everybody's role is really equally important. I don't want to say that one is less
Yeah.
important than the other, but
Yeah.
um, there's a lot of reasons and time points in which studies can go sideways, and it's very, very challenging. And that's what I love about this work. It's really just doing the impossible, what people perceive to be the
Right.
impossible, which is not.
Right.
But that's why I just like on my recent post on LinkedIn, it's like I really wonder how many people really truly understand the depth of what we actually talked about today. And we've been very high level too, right?
Right. Right.
My latest post is like, it's around just public misunderstanding of research. And it's not
Of
like
course.
our industry are in some underground bunker, corroborating some grand scheme and we're lying to people. No, no, it's actually not. That's so far from the truth. And so that's what I mean by, it's a very complicated process, and explaining this to people who aren't insiders, they don't, is challenging. And so, investors oftentimes will look at risk and success and likelihood of market, commercialization, and it's tough. But I think investors need to, without the investors, there is no
Oh yeah.
development.
Yeah.
And so I encourage all the investors out there to keep investing, create
Well,
those funds, and they do.
yeah, there's money out there. It just depends what it's going for. But we appreciate your time. We're
No, of
gonna do
course.
more. Maybe we'll do a series of these because I think we are in the golden era of biotechs, just like we are in the golden era of sites. Maybe those two things go hand in hand. I got Joel White on Friday. I know you're gonna
Oh,
want to
nice.
watch, and I apologize for my upload speed on YouTube. It's gonna be good because it's saving both local files. But for those watching live, hey, you got it first, but the upload speed on my end's pretty bad. We had a storm here. Joel White is gonna be on discussing the quarter 3 CRO analysis. It's pretty interesting, man. It's looking like they're getting creative as far as how they're getting their revenue, and it's not the fundamental way that their business was supposed to get
Yeah,
revenue. So we can talk about that at some point, but
definitely.
I think it's not the golden era of CROs, is what I'm trying to get at. But sites and biotechs, I think they're kind of going down the same path, and I think they both need each other, even though they don't really interact without the
Exactly. And
CRO.
I think that's a subliminal message for what's to come, right?
Oh, for sure. I mean, thinking about it, yes, it's not what I meant, but yeah, it's
I know,
reality.
right? But I'm just saying,
Yeah,
like,
yeah, yeah.
that's
No, you're
yeah, you're
absolutely right.
no, you're
Absolutely
spot
right.
on too.
We got
So
things coming, guys. All right, everyone, go follow Robert Goldman, the sponsor monster. This dude, way too talented for LinkedIn. You need to be on X, man. I
Yep,
need to
I'm
get
I'm
you on
I'm
next.
heading over there. I'm heading
Okay.
over there. Thanks, man. Appreciate
Like,
you very much.
subscribe, comment, share. Thank you everyone for watching. Bye bye.
Bye bye.
Bye bye. Bye bye.